In this video, we will discuss about multiplexing results of 8x NFAT reporter signals. So basically, the researchers wanted to check if there are multiple T cell lines exist at the same time, whether it will affect their NFAT reporter signals, or how reliable is the NFAT reporter signals when there are multiple T cells are present in this sample. So in order to check that, what it is, they use two different fluorescence combinations for each cell lines, and then first they check the resting ZS green signals, and they can see that in most of the cases the resting ZN green signals is lower, except for the one case where the percentage ZS green signals is higher, and these results are for the unstimulated T cells or non-activated T cells. However, the researchers said that this increase is also within the range to distinguish between activated T cell signals and non-activated T cell signals. Thereafter, these signals are measured once these T cells are co-cultured with autologous B cells, but that does not have this antigens to activate these T cells receptors. Next, they co-culture it with these peptide concentrations or the insulins that can help in the activations of these T cells. And what they are able to see that even at the lowest concentrations of around 0.1, I think it's microgram, and they are able to see some signals. Or we can see that in this figure in the panel B, they can see that with the relative increase in the peptide concentrations, there is a relative increase in the signals, and this indicates that even in the case of multiplexing or the multiple signals, there is a relationship between this increase in the peptide concentrations to the ZS green positive signals. And here this stimulation index represents the signals ratio of the signals for the stimulated conditions to the resting conditions, and we can see that it increases with increase in the peptide concentrations. That is what they are expecting. And thereafter, they wanted to check the changes that they see when the single fluorescence channel is activated compared with the multiplex signals that is activated. And when they did it for like in all the 8 fluorescence combinations, they are able to see that in all the cases the signals that they observed from the single fluorescence reporter compared to the multiple fluorescence reporters are almost same. That can be seen here. Now they also wanted to check whether these activations, when there are multiple T cells are present, whether their NFAT reporter signals get activated because of the activations of the another T cells, or it is activated because of the antigens present within the medium, cell culture medium. So in order to do that, they used different combinations, and here they have shown the combinations that for example this in the panel D for the first figure, when they use the AM plus BFP, they can see the signals for the AM plus BFP, but not for the other fluorescence channels because these are not been activated. And we can see that their signals are 0 even at the different peptide concentrations up to the 100 microgram per ml. And similar results they have seen for all the 8 different fluorescence combinations. So this also indicates that the multiplexing, this NFAT reporters gives the reliable results even for the multiplexing or when the multiple T cells are present, and these are not affected by the activations. These signals are not affected by the activations of another T cells, but are the results of the activations, but are the results of the actual activations because of the peptide or the antigens for which the T cells are designed or are sensitive. So that's all for this video. I hope you understood how they check their multiplexing results of their 8XNFAT reporters. If you need to understand more about this experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.